Imagine a compound that reduces inflammation as effectively as some anti-inflammatory drugs in laboratory conditions, that kills cancer cells in petri dishes without harming healthy cells, that protects the liver from toxic damage, that crosses the blood-brain barrier and protects neurons, that has antimicrobial properties, that has antioxidant properties stronger than vitamin E. And imagine this compound has been consumed daily by hundreds of millions of people for 4,000 years with no documented toxicity. This is what early laboratory studies of curcumin, the active compound in turmeric, were finding in the 1980s and 1990s. The results were extraordinary. Researchers were genuinely excited. And then, the clinical trials happened, and things got more complicated. The first serious Western scientific attention to turmeric came from Indian-American researcher Bharat Agarwal at MD Anderson Cancer Center in Texas. In the 1990s, Agarwal began publishing studies on curcumin's anti-cancer properties. His work showed curcumin suppressed NF-κB, a protein that plays a central role in inflammation and in the survival of cancer cells. Turn off NF-κB and cancer cells became more vulnerable. Curcumin appeared to turn it off. Agarwal's papers attracted enormous attention. He became one of the most cited cancer researchers in the world. Curcumin became one of the most studied natural compounds in cancer research. And then, in 2012, Agarwal's papers were investigated for data manipulation. Multiple papers were retracted. The story got complicated. But the interest in curcumin his work generated has never gone away. The problem with curcumin in clinical trials comes down to one word: bioavailability. Curcumin is not well absorbed by the human body when consumed in standard form. You can eat enormous amounts of turmeric and very little curcumin actually reaches your bloodstream. Most of it passes through your digestive system without being absorbed. This is why the laboratory results did not translate cleanly into clinical results. In a petri dish, you can expose cancer cells directly to curcumin. In a human body, the curcumin has to survive the digestive system, get absorbed through the intestinal wall, reach the target tissue in sufficient concentration. And it is very bad at doing all of this without help. This is the bioavailability problem, and it is the central challenge of all curcumin clinical research. The ancient Indian solution to the bioavailability problem is piperine, the compound in black pepper that gives it its bite. When curcumin is consumed with piperine, bioavailability increases by up to 2,000%, not 20%, 2,000. A 1998 study in Planta Medica confirmed this with clinical data. Piperine inhibits the enzymes that break down curcumin before it can be absorbed. It effectively blocks the body's own mechanisms for eliminating curcumin too quickly. And remember haldi doodh, turmeric milk, the ancient preparation drunk by Indian families for centuries. It contains black pepper. It contains fat, which also improves curcumin absorption. The ancient formula was already solving the bioavailability problem 2,000 years before science named the problem. The recipe was the answer, written before the question was asked. Modern pharmaceutical research has now developed enhanced curcumin formulations: nanoparticle curcumin, liposomal curcumin, curcumin bound to piperine, phytosome, technology that binds curcumin to lecithin for better absorption. All of these improve bioavailability dramatically. And when curcumin actually reaches the target in sufficient concentration, the results are more interesting. Clinical trials on curcumin-enhanced formulations in osteoarthritis show significant reduction in pain and inflammation comparable to ibuprofen. With no gastrointestinal side effects, which ibuprofen causes in many patients. Early trials in colorectal cancer prevention show promising results. Trials in Alzheimer's prevention are ongoing. The compound that was dismissed as folk medicine is being taken very seriously by serious pharmaceutical researchers. Why did it take so long for Western science to take turmeric seriously? Several reasons. Funding. Basic research into natural compounds does not generate pharmaceutical patents. If curcumin works, it cannot be patented. If it cannot be patented, there is limited commercial incentive to fund the trials that would prove it works. This is a structural problem in pharmaceutical research. Not a conspiracy. Just the logic of how research funding works. Studies of unpatentable natural compounds rely on government funding and academic interest, which is slower, which is less funded, which means the research that might confirm or disprove traditional knowledge takes decades longer than it should. The 4,000-year-old Indian grandmother was right about turmeric. And the proof took this long partly because the truth was not commercially convenient. What the lab found? Extraordinary early results. A bioavailability problem that explained why the results did not translate directly into clinical success. Ancient formulations that had solved the problem before science identified it. Enhanced modern formulations now producing clinical results that are genuinely promising, and a structural problem in pharmaceutical research that delayed the proof of what 4,000 years of use had already demonstrated. Turmeric is not a cure for cancer. That is not what the science shows. That is not what Ayurveda claims. Ayurveda claims it is a powerful tool for maintaining balance and preventing the conditions that allow disease to take hold, and the science is beginning to confirm, that there is something real in that claim, something worth taking seriously, something 4,000 years of Indian grandmothers already knew.